but we don't. BRCA1, BRCA2, we don't check the tumors.
We check.
We don't check the family.
Yeah.
We check to check. So BRCA1, BRCA2 will tell you if the patient is high risk before developing the tumor. She doesn't have a tumor. We will say that person has high risk of tumor or not. But HER2, we check it when the patient has tumor, has cancer, to see if the patient responds therapy. واضح? So that's the difference. So these are the two major differences. Now, so if the patient has breast biopsy for breast cancer, I send these for amplification study using fish. If I have lung cancer, I send a piece for sequencing for PCR, looking for point mutation, PCR study, molecular study, point mutation for lung cancer. For HER1, or we call it EGFR. It's better to be called EGFR, but it's called HER1 also. HER1. So this is the only thing I wanted to remember. Point mutation in lung cancer and amplification in breast cancer. And these are important because they are predictive markers. What are the predictive markers? They predict the response to therapy.
Yes.
So those we can give a targeted therapy.
So that's HER2?
HER2 and EGFR. HER2 in breast and EGFR in lung and in carcinoma.
Yeah, but if she already has cancer, but don't you sometimes check for BRCA just to make sure that her family might be at risk?
Yes, I check, but this is blood.
Okay.
Yeah, the patient comes, if the patient is young, and in the history, her grandmother, sister has breast cancer, then I say, I'll take blood to check. If she's positive, then I'll bring that breast. So the blood, that's the thing.
But for her sake, you check her too.
But if she has cancer now, BRCA1, BRCA2 for her is not risky. She has cancer already.
Okay.
So, but I'm checking the BRCA1, BRCA2 in her to see the family.
Okay.
The risk of the cancer. The family. No, BRCA1 indicates the risk of cancer. HER2 is the for medication. And for lung cancer, EGFR. And for colon cancer, what? KRAS. We said
KRAS to check.
KRAS for
These are often all ABC to check.
All mutations you check.
ABC familiar, yes. ABC familiar. So, this is the HER2. Again, the treatment for HER2, we have Herceptin. So I'll give you an example of what we are saying now. So the patient has the growth factor, which we don't know, HER2, we call it orphan, unknown.
Yes.
So stimulate the receptor, and the receptor is tyrosine kinase receptor in this one. The KRAS we say is G protein. G protein in KRAS. But in HER2 and in EGFR, all EGFRs are tyrosine kinase receptors. EGFRs are tyrosine kinase receptors. The tyrosine kinase has three receptor, transmembrane, and tyrosine kinase domain. So they will cause amplification of signaling, transduction signaling, and cause the cell to proliferate. In 20% of breast cancer, and those that have HER2 amplification, we give this medication, Herceptin. And Herceptin, it will block the receptor. It will block the receptor, so it won't be active.
So HER2 and EGFR are both tyrosine kinase.
All EGFRs are tyrosine kinase. We have 4 EGFRs. Here, 1, 2, 3, 4.
Oh, okay.
All the EGFRs are tyrosine kinase receptors.
Yes.
All the EGFRs. HER2 is EGFR. It's one type of EGFR. So 4 EGFRs. EGFR 1, and here 1, 2, 3, 4. And all of them tyrosine kinase. Whereas the KRAS are the
G protein.
G protein. خلاص انتم صرتوا علماء في الان تطلعون على العمل راسا. Now we spoke about molecular mutation. ركزوا معي شباب. We spoke about molecular mutation, and we say the two critical genes, oncogenes and tumor-selective genes. Now we are speaking about two more genes that are very important and recent, and there are so much research. They are clinical trials, but we don't have trials. We don't use them in diagnosis. They are the future of medicine. The epigenetics and the microRNAs. So these are the two. I will briefly talk to you about them so that you know what we are, because these are the future of medical medicine. And epigenetic and microRNA, they cause the silencing of gene. What's silencing of gene?
Coiling maybe to the parts of the chromosomes.
RNA silencing.
Yeah,
RNA interference.
What's silencing? Yeah,
You interfere the coding so it doesn't duplicate the mRNA.
Won't be
not expressed. The gene is not silencing, so epigenetic and microRNA cause silencing of gene. شباب ركزوا معي. Silencing of gene means the gene is not expressed. When we say silencing of mutation, means the protein is not changing the protein, right? When there is silencing of mutation silencing means the protein, the end result of the protein is not changing. But silencing of gene, the gene itself is not expressed.
Yes.
So the epigenetics, modification of histones, and DNA methylation. And this DNA methylation, if there's DNA methylation, the gene of that gene, it won't be expressed. The commonest one, it won't be expressed. The commonest is in the promoter region of, there's hypermethylation of promoter region of tumor suppressor genes.
So there's no tumor suppressor gene.
Yeah, it's not expressed, inactivated. So the three tumor suppressor genes, the commonest where we have epigenetic phenomena are the BRCA1, the breast cancer, VHL, I will talk about cell carcinoma, and MLH1 colorectal.
So when we check for BRCA1, we're checking if it's not there.
No, we said the tumor critical gene, or oncogene, that it should be activated for cancer to happen. And tumor suppressor gene, it should be inactivated for tumor suppressor. The inactivation of tumor suppressor gene by two ways. Either mutation of the genes, or silencing the through epigenetic.
Which is, that's called epigenetic change?
Epigenetic. Silencing. Hypermethylation of the promoter region. You know, promoter region is not part of the gene.
So methylation or mutation?
Sorry?
Methylation or mutation?
Yeah, there's two.
Two mechanisms. Mutation in the gene, hypermethylation in the promoter region.
And hypermethylation is silencing.
Silencing. It's epigenetic. واضح شباب؟ So tumor suppressor gene are inactivated. For in cancer, tumor suppressor gene has to be inactivated. And inactivation by mutation in the gene or hypermethylation of promoter region. Promoter region, you know, it's not part of the gene. Yeah. So promoter region is hypermethylated, the gene is silenced, is inactivated, and that's the goal in tumor. So tumor cancer. And that's what we call epigenetics. So epigenetics is hypermethylation of the promoter region in the tumor suppressor region.
Can you explain a little bit more about what's hypermethylation? What's hypermethylation?
Hypermethylation, more methyl group, it will be attached to the promoter region. Methyl group, it's not present, so it will be attached to the promoter region, so it silences the gene. The gene won't be expressed. The other thing is the microRNAs. So anybody knows what microRNAs are? أخذتوها? Normal. It's normal in the cell microRNAs.
Yeah.
So in microRNAs are, you know, the central dogma of molecular biology. You have gene, messenger RNA, and protein. Now there are in the cell multiple small RNAs. We call they are expressed from gene. We call them microRNAs because they are 2022 nucleotides. Short, and they are attached to the messenger RNA and they silence the expression.
So microRNA attached to messenger RNA.
To silence the expression.
So you can make it synthetically.
So the tumor suppressor genes attached to the microRNA.
Yes.
So there's cancer.
Yeah. So if they target oncogene, they become what? They become tumor suppressor gene like. If they target tumor suppressor gene, become oncogene like behavior. So microRNA small segment of RNA that is expressed by the genes, normally regulate the gene function through silencing of some gene expression. Normally, they silence some gene, but this is normal function. Like the tumor suppressor gene, oncogene, they are normal, right? They control the cell division and cell growth. MicroRNA also regulate the whole genome function. So it silences, it stops the expression of this gene, express this gene and so on. But when they become mutated, then they become like oncogene or tumor suppressor gene. If they attach to oncogene, it's acts like tumor suppressor gene. And if they attach oncogene, yeah, you are.
Yeah,
but doctor, how can they normally attach to RNA?
Yeah.
But tumor suppressor genes are DNA, so how can they?
The tumor suppressor gene are DNA, but they have to come protein. They have to
come from RNA.
So
But their mRNA is expressed gets and their proteins cannot be produced, which affects cell cycle.
the silencing is not as in the
normally they are normal.
Yeah.
But when they are mutated, they might not get
So beside the tumor critical, so we spoke about
they cause them to be
apoptosis genes, which are important. Tumor suppressor gene, tumor oncogene, and epigenetic and microRNA. These are the future of the molecular genetics. I just want you to understand the concept, not the details.
I think it's going to promote.
So this is the multi-step. There is no one mutation that will cause cancer. I told you that before.
Just the microRNA, you said it silences the tumor suppressor gene, and what does it do to the oncogene?
Okay, silence tumor.
So
So the silence oncogene, it will become behave like tumor suppressor gene. The silence tumor suppressor gene behaves like.
But that will not cause cancer.
Yes, it will cause cancer.
Oncogene, we just said that it's going to act as a tumor suppressor gene, so that's not going to cause cancer.
No, I'll explain that to you alone. I'll show it to you. So let's move on because of time. You have tumor suppressor. You have this multi-step process. Shabab, let's focus. Multi-step process. You have from normal colon to cancer. Multi-step process. Not one mutation that will happen. Shabab, focus on that. You have the mucosa, submucous normal. The first is the APC. This is established multi-step process of carcinogenesis. Multiple tumor suppressor gene and oncogene will happen. So start with the APC gene, either germline or somatic. We spoke about that mutation. Then you have mutation in the activation. This first hit, the second hit. Remember that tumor suppressor gene. We have 200 Then we have KRAS mutation, the proto-oncogene mutation. Then we have other mutations in tumor suppressor genes, and so on. So, and as we have mutation, it changes from normal colon to adenoma. Adenoma means polyp. The polyp that we saw with dysplasia. Then become carcinoma, then invasive carcinoma. So you have multiple steps, not one mutation. And that's why you have to understand this concept. When you have 20% of cancer patients have HER2 amplification breast cancer, you give them the therapy, and after time they get resistance because you have other genes that are working. You have 20-30% of lung cancer, they have EGFR mutation. You give them therapy. After a few months, 1 year, they become resistant because you have other mutation that comes.
They cause mutation.
Other mutation that will drive the growth of the tumors. So it's not one mutation; it's multiple mutation that happens. But you want to see the mutation and act. The scientists they want to know what starts the cancer, what's the beginning mutation, and what are the driver mutations that will make the cancer continuous, driver mutations, and the beginning mutations. So the cancer is really multi-step process, is not one mutation. Tumor suppressor gene, first hit, second hit, oncogene, tumor suppressor gene, and so on. It goes. And different stages, yes.
So that's why when you are giving antibodies or target therapy, you should give him chemotherapy with it.
No, you don't have to. I mean, they are with you. Sometimes you give it, sometimes you don't give. But chemotherapy, It is not selective. Any cell that proliferate, it will act. It's not selective against that cancer. Any cell in cell cycle, it will act on it, the chemotherapy. Target therapy, it will act only on the cells that have mutation.